Welcome to lesson 5. We are moving on today to have a look at kidney structure. So we're going to have a look at a dissected kidney, or at least a picture of one. We're going to have a look at the internal structures, and then we're going to have a look at a nephron, which is the filtering unit. A nephron is one filtering unit, and you have approximately 1 million to 1.5 million in each kidney. So where are the kidneys? They are in your abdomen. If you put your hands on your waist, then your thumbs should be able to go around to roughly where your kidneys are. You've got one either side of your spine. They are at the back, so you have your stomach and your liver in front of them. The left one is ever so slightly higher than the right one. And then you have these strange things on top of your kidneys. You can see them here. And they are your adrenal glands. Now, adrenal glands we will be coming back to when we look at hormones in year 13. Anything renal is kidney. So when we looked at the liver, it was all hepatic. So hepatic artery, hepatic vein. The kidney, it's all renal. So you have the renal artery taking blood into the kidney. So have a look at the diagram, see if you can find renal artery, and you should have two of those. They each kind of serve one kidney, and they branch off the aorta. And then you've got two renal veins taking blood away from each kidney, going back into the vena cava. Now, the blood going to the kidney is obviously going to be richer in urea. During the ornithine cycle in the liver, you made urea. That urea will be chucked out of the liver into the blood, or will leave the liver in the blood. That blood will circulate to the kidney, and the blood in the renal artery taking blood into the kidney will have more urea. The kidneys, the nephrons, will filter out that urea, and so the blood leaving the kidney in the renal vein will hopefully have less urea, and if it doesn't, there is something wrong with your kidney. So leaving each kidney, you have something called a ureter. The ureters leave each kidney and drain the urine, the newly formed urine from your kidneys, down into your bladder, and then the urine leaves your bladder through the urethra. Now, ureter and urethra are two terms that students get the wrong way around. It is alphabetical, just ureter comes slightly before urethra in the alphabet. Okay. Right, so this is what a kidney looks like inside, and there are some terms that you need to know. So the renal capsule, that is a membrane, a transparent, very thin membrane that goes around each kidney. You've got the renal cortex, which, if I try to highlight it on here, it goes all the way, it's this outer edge, it's this bit. Now on this diagram, this kidney, it looks a little bit lighter. It should actually be darker, and if it was a fresh kidney, it would look darker. That's the cortex, that is the outer portion, and it should look darker, and you'll see on the next slide it is darker if it's a fresher specimen. This one's been preserved. It should be darker because it contains knots of capillaries called glomeruli. Glomeruli is plural, glomerulus is one. Just inside that, you have something called the medulla. So again, it's around here, and then it's this area. So this is an area. It is that kind of middle area. So it's the medulla there. Within the medulla, you have pyramids. So you can see here, that's one pyramid, that's another pyramid. Now these pyramids are actually three-dimensional structures. So if you were to look at one, sorry, that's a terrible drawing, it would kind of look a little bit like that, probably a little bit flatter at the end. So it kind of looks a little bit like a piece of sweet corn. So that's one pyramid. This would be one pyramid, and you can see you've got 1, 2, 3, 4, 5, 6, 7, 8 or something that you can see there. And they are in that area called the medulla. So the pyramids are in the medulla. And then the rest of it, all of this white stuff here, that is the pelvis. And it's kind of a funnel shape because coming out of the medulla is urine. You've got all these collecting ducts and the urine is flowing down and it's picked up in this pelvis. And the calyx is kind of, it's these cupping, let me get rid of everything else. The calyx are kind of these cups that go around the bottom of the medulla, of the pyramid, sorry. And it's collecting that urine, and then that urine drains into the pelvis and then leaves the kidney down the ureter. Okay, now the renal column, which is here, is just this area between the pyramids. Let me try and highlight that a little bit better. So it's this area between the pyramids. It's a column of tissue in the medulla that's between these pyramids. It's not something we dwell on too much. So on the next slide, we're going to have a go at drawing one. So you need to be on page 20 because you have a torso to label. So you can have a go at labelling the bits and pieces associated with the kidney, and then we're going to draw a kidney. So on the right there is a dissected kidney. That is 1/2 of a dissected kidney. So it's been cut, sliced along its length and cut into, so it's a longitudinal section. And what you can see very nicely here is this area, which is the cortex, which is this darker stuff around the outside. It is rich in capillaries. That's not a very good, so that's the kind of the border, if you like, between the cortex and the medulla. So this is the cortex, and then if I was to draw a line around there to separate off that bit, that would be the medulla. So the medulla is all of that. Okay, so this whole area, if I was to crosshatch all of this, this would be the medulla. Okay, so it's an area. Just get rid of all of that. Now within the medulla, you've got these structures, oops, which are the pyramids. So you've got one there, you've got one there, you've got one there, and you can see that calyx round there draining down into this pelvis, which is the white stuff. Okay. Anything else to note there? And so this area between them, between these pyramids, that would be the renal column. It's just part of the medulla, not the actual white stuff. So this would be the pelvis, this would be a pyramid, this would be the cortex, and this would be the medulla. Okay, so you are going to draw this kidney. Like I said, the one on the left here is my drawing of this kidney, but you need to make your own drawing. So have a go at doing that. Please do not just copy mine. You are trying to develop your drawing skills. Obviously, this diagram here is very, very badly labelled, and I didn't just label mine very well. So please use those rules of drawing and labelling, nice crisp lines, labels going out to the side. So do your drawing nicely in the middle of that available space, so you can put labels on either side so you don't crowd your drawing. They should have horizontal ruled lines, and then labels should be either in pencil or pen. No arrowheads. Have a go at drawing that. Okay, so pause it for a minute and then have a go at drawing that, and then you can have a go at the gap fill on page 21. So it's talking you through the kidney, and then it touches on the nephron. Each of those filtering units, those units that you have inside of these kidneys that you can't see, obviously, when you just cut into the kidney, that is a nephron, and that's what we're going to spend the rest of this particular PowerPoint having a look at. So pause, have a go at drawing that, and then continue. Right, so this is a nephron. Now you have seen this before at GCSE. The diagram on the right is the one that you have in your book on page 22, I think it is, and you have some labels to add to from that, so you can use the labels on that right hand diagram to try and identify all those bits. There are a couple of things that I want to point out. So here it says arterial from renal artery, arterial from glomerulus. I want you to use these terms, afferent and efferent. This one is the afferent, this one is the efferent. Afferent goes in, efferent comes out. So let's trace the fluid, if you like. So the fluid comes in as blood into the afferent arteriole. Now the lumen of the afferent arteriole is bigger than the lumen of the efferent arteriole because you want to create pressure, you want to generate pressure, because as the blood comes in, quite a lot of the fluid and the substances dissolved in it are pushed out of the glomerulus and they are pushed into this space. Can you see this space between this yellow space? That is Bowman's space. So this knot of capillaries here is called the glomerulus, and then that kind of cup-like thing surrounding it is Bowman's capsule. You can see Bowman's capsule over here. I'm just rub that out. So you can see here is Bowman's capsule. Now these diagrams are, it's very, very difficult to show what the relationship between the glomerulus and Bowman's capsule, because it looks like there is space between the two. Now it is true that if I try to draw Bowman's capsule like that, and then that goes off to that tubule, and then the network of capillaries, be easier if I had a different colour, comes in here, you get that knot of capillaries, and then you get the efferent arteriole leaving. It looks like there is a space sort of here. That isn't a space, and I'm going to try and draw it out for you to show you. Now this is a space. So anything that is pushed out of the blood gets pushed into this space. Okay, and then eventually it will work its way down this tubule. It is then going to form urine eventually. So let me try and draw you the relationship between Bowman's capsule and the glomerulus. So if I draw a capillary, so that is the capillary. Let's colour it in so you know it's a capillary. Now the inner kind of part of the glomerulus actually has very specialized cells that have these projections that kind of look a little bit like fingers, and another cell will have projections that kind of interlock, and so they are wrapped right the way around this capillary, and so there is no space between the capillary and the glomerulus. Anything that leaves the blood is picked up into this glomerular space, and then the rest of that capsule will be all the way around it and go off into that proximal convoluted tubule. So there is no escape for anything that gets pushed out of the blood. It will be picked up into this Bowman's capsule, into this space that's kind of there. It's that yellow stuff there, and then eventually once it's pushed out it will then flow through these tubules. Okay, so you've got afferent arteriole going in, efferent arteriole coming out. The knot of capillaries is the glomerulus, is one, glomeruli is many. That's picked up by the space, the Bowman's space in the Bowman's capsule, this thing that's kind of wrapped all the way around that glomerulus. And then that fluid is now known as filtrate, and it works its way through the first part of this twisty tubule. Now, because it is a twisty tubule, let's get rid of all of this for a second. Because it's a twisty tubule, it is actually called a convoluted tubule. I pick the wrong colours. It's called a convoluted tubule. Now, because this one is closer in proximity to the Bowman's capsule and glomerulus, it's called the proximal convoluted tubule. That fluid then goes down the loop of Henle, down the descending limb of the loop of Henle, turns that bend and goes back up the ascending limb of the loop of Henle, and that should have a capital H. And it then goes into the distal convoluted tubule. Again, it's a really twisty tubule, so it's called convoluted, and this one is further in the distance, so it is the distal convoluted tubule. And then that urine drains into something called a collecting duct. Now you can see here you've got more than one kind of entry point, and that's because, let's get rid of all of this. So if I try and point to those a little bit more, you've got one there, you've got one there, you've got one there, one there. That's because more than one nephron can drain into each collecting duct. Collecting ducts are a bit larger. You can have urine formed by all of these nephrons, and several nephrons can drain into each collecting duct. So you have fewer collecting ducts than you have nephrons. Okay. Now the other thing to notice on this diagram is you can see here, let me get it, the efferent arteriole, when it leaves the glomerulus, it then wraps itself around the loop of Henle, the proximal convoluted tubule, the distal convoluted tubule. You'll see on the next slide there's slight differences in the wrapping of the efferent arteriole, because anything that comes back out of this filtrate, so obviously you formed the filtrate going in, but anything that's reabsorbed back out has to be picked up by the blood. So you have to have a good network of capillaries wrapping around these tubules. So anything that is reabsorbed into the blood, and eventually this will go off and drain back into the renal vein and leave the kidney. Okay, so like I said, this one over here is the one that you've got in your booklet, and you can have a go at labelling it. If any of you get stuck, you can all take a photo of it and send it to me, and I'll tell you if you've got it right. Okay, so like I said, there's approximately 1.5 million of these nephrons in each kidney. Now you can see here this area, let me try and get the right colour pen this time. This area here is the cortex, and you can see it's because you've got all of these glomeruli. So you've got these knots of capillaries, lots of blood, and so that's why they look darker in colour. That area generally looks darker in colour, and you can see you kind of get a slightly stripy look to the medulla because of all of those ducts. You've got all of those loops of Henle dip. So the proximal convoluted tubules and the distal convoluted tubules are in the cortex. The loops of Henle and the collecting ducts are in the medulla. So have a look at that. Make sure you can find the proximal and distal convoluted tubule. Make sure you can identify the descending and ascending limb of the loop of Henle and the collecting ducts. Now this is a slightly easier version. So if you were drawing this out for revision, I draw something that is actually even more simplified than that. So I would draw the afferent arteriole, the glomerulus, the efferent arteriole. I would draw Bowman's capsule. I would just do a wiggle for the proximal convoluted tubule, draw a loop of Henle, doesn't matter how long, draw a wiggle for a distal convoluted tubule, and then a collecting duct. Now obviously you can draw one that's slightly better than that, because you're not trying to draw it on a screen with your finger. Okay, so that's the kind of thing that I would use for revision, not those very, very tricky, weird things. Now, you do need to be able to identify the different parts of a nephron. You do need to know the difference between the proximal and distal. You do need to know what the glomerulus and the Bowman's capsule and all those different bits are. And you have space on page 23 where you can do the structure and function of the different bits of the nephron. And it even tells you using page 421 of the A2 textbook which is the one that's on Caboodle. Now this diagram, you'll never be given something like this, it's far too complicated, but this is just to show you if you are doing any kind of research, if you are looking anything up, there are two different types of nephron. The version we've just looked at is a juxtamedullary nephron. There is also something called the cortical nephron, and you can see immediately actually they have slight differences in their loops of Henle and where these capillary networks are. It's not something you need to worry about, but it's just there just in case you come across it and you were, I don't know, interested. Right, so as it says there, make notes in your workbook using the textbook on Bowman's capsule, proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting duct. Okay, so here's a nice little quiz for you. See if you can identify these bits. So if you want to pause and have a go, and I will go through it in a minute, but I will just give you a chance to pause, try to label them, and then you can come back. So just pause it now. Right, so hopefully you've had a go. Now I'm just going to write in the initials because I can't write in these tiny weeny little boxes. So let's have a look at the first one. This one is the glomerulus. It's a terrible G. The next one, shall I do it in a different colour so you can see it better? The next one, that is Bowman's capsule. And you can see that space between, so that would be Bowman's space. If you want to label that, you can. This one would be the proximal convoluted tubule. It's proximal to the glomerulus, it's a bit closer. This is the loop of Henle, and this particular part is the descending limb, and then this, that bit over there would be the ascending limb. Then we go into the distal convoluted tubule, then we go into the collecting duct, and then this would be the urine to the ureter. And then eventually, obviously, they'll drain into the bladder. Okay, so hopefully you got all of those right. I hope that you can actually see that. And thank you for this lesson.